Okay, so let's talk about mitosis. Mitosis is how we make clones of cells. There is no switching up of genetic material, there is no mixing of genetic material. It literally is, I'm going to make a carbon copy. So, in mitosis, we basically have two major phases. We have interphase. Inter, that prefix, means in between. I don't know why that's doing that, but it means in between. And then you've got the mitotic phase. So during interphase, this would be like going from being born to being reproductively active. Okay. Basically, you are just living everyday life and growing and getting things ready inside your cell so that if you do decide that you want to divide, you can. The mitotic phase is the one that we hear the most about, but to be honest, it's not a huge part of a cell's life cycle. In fact, it's probably less than a quarter of it. So, the cell cycle, like I said, you've got interphase, which is about 7/8 of a cell's lifetime. It's almost its whole life. And it's the time frame between cell divisions. Mitosis is that 1/8 that's missing from interphase, where a parent cell produces two daughter cells. And remember, those daughter cells are clones. They're not any different than the parent was. So let's go into interphase. Interphase starts with G1. So as soon as I'm done dividing, I go into G1 phase. The cell is metabolically active. I'm making energy. I'm making proteins. It duplicates its organelles and cytosolic components. In other words, it duplicates its mitochondria, its ribosomes, its Golgi, its endoplasmic reticulum, and it'll start replicating centrosomes. Okay? Now, that is if it's going to go back around to do a mitotic phase. If it is not, it goes into something called G naught. That little O right there, we say it means not. Basically, what that means is I'm not going to divide. I'm not going to change anything in my cell. So I'm just going to kind of go into living everyday life, but I'm not going to do anything else fancy. So during G1, growth phase 1, it occurs when the cell is first formed, right after that division point. The cell is metabolically active. It's making copies of its organelles and any other things that need replacing in its cytoplasm. Let's say that I've got a lysosome that isn't doing so well, so I need to get or make a brand new lysosome. Sorry, my glasses are really dirty. And basically, it's just daily living. I'm just living. I'm doing my metabolism. I'm making energy. I'm making new proteins. Just daily living. After that, you have interphase. Again, that's if you decide not to go into G naught. So during S phase, we call this the synthesis phase. Okay? The DNA is replicated at this point. Okay? Why? Well, if I'm going to make two clones of myself, each one needs to get a copy of the instruction manual. So I do need to replicate my DNA so that each one gets their own whole book of instructions. Finally, we go into G2. Whoa. In G2, or growth phase 2, the cell continues to grow until it is twice as big as it originally started. So remember, if I'm going to divide into two daughter cells, they have to be, once I've divided, the same size I was when I started. If not, every subsequent division, they would get smaller and smaller and smaller until we just didn't have anything left. Other proteins are also going to start getting made here. Okay, why? Because we need those proteins to help with the moving and the shaking and the making extra membrane and the splitting and all of that. Something else that happens during G2 is the replication of the centrosomes. Now remember, the centrosomes are the things that move the DNA around, or the chromosomes around, so that when I do decide to go through my division, they actually split equally. One set of instructions going to one daughter cell, one set of instructions going to the other. So this is a model of interphase. You can see chromatin in the nucleus. You can actually see the nucleus, which is another big hint that I'm in interphase. You've got cytoplasm, you've got the cell membrane. If you look here, you can see that the centrioles have been copied. I've got two black dots. If you look as well, you can see that there's kind of this starburst pattern here, right next to those. That's actually called the aster. What's the symbol for the astros? Stars, right? So that's what the aster looks like. It looks like a starburst. Now looking at the mitotic phase. You can think of the mitotic phase, if you want to have something to kind of remember PMAT. Okay? Prophase, Metaphase, Anaphase, Telophase, PMAT. During mitosis, the cell is actually dividing into two new daughter cells. The first thing that really happens in mitosis is the division of the nucleus. The nucleus itself does have to divide and, excuse me, split the DNA in half. The second part, kind of, I guess, the thing that we visualize when we see a cell splitting is going to be the cytoplasm being split in half, where one daughter cell gets half the cytoplasm and the other daughter cell gets half the cytoplasm. Now, that process, I'm sorry, I'm cleaning my glasses, please forgive me, but they are so dirty at this point, I can barely see. Okay, back to my pen. The cytoplasm being split is called cytokinesis. Cyto means cell. Kinesis is to split. Okay? So cell splitting, literally the word tells you what it means. That's one of the reasons why I always say if you learn what the meanings of things are, it can kind of help you. So you've got the interphase in G2 here. The first one, remember, PMAT. Okay? You've got prophase, prometaphase, metaphase, anaphase, and finally telophase and cytokinesis. So, we've got the stages on this slide that we just showed on the last slide. Let's talk prophase. During prophase and prometaphase, kind of the first two time frames, one of the things that you are going to notice is that genetic material starts to get very dense and thick. Instead of being really spindly like thread, it starts becoming more dense, which by the end of prometaphase, you can really see, okay? Something else that starts happening, that nuclear envelope starts to disappear. You don't see it anymore. It starts to break down. Why? Well, the nucleus keeps everything inside, but I'm going to divide these cells and half needs to go in one direction, half needs to go in the other. It can't be contained in the nucleus. It needs to get that out of the way so that when I split those apart and divide half and half, Half goes one direction, half goes the other. As I said, the chromatids, which is the unraveled kind of thready DNA, start to condense into chromosomes, and they have copies. So if you look here, it looks like I've got something like this attached to something like this. Each one of these is a copy. So if this was chromosome 1, this would be copy A, and this would be chromosome 1, copy B. They're the exact same thing. It's the exact same instructions. It's just that they're attached to each other. So they're attached to each other by something called a centromere. Okay? This little area where you kind of see that little indentation, that's the centromere, where they're attached to each other. Okay? Now, this little black thing on the outside is actually called a kinetochore. Remember I told you that our centrosomes are going to pull and push and move them where we need them to go? This is actually where they attach. Now, as prophase starts, you'll notice that that aster, that starburst I told you about, starts to kind of look like it's making lines here. And these copies of our centrosomes are starting to get pushed side to side. That's actually really important. That is called the spindle. See how this kind of looks like an almond shape here? That's actually what spindle means. If you've ever read or seen the cartoon or the live action movie with Sleeping Beauty or Maleficent, she'll prick her finger on a spindle, right? Well, that spindle they're talking about is actually this huge needle. And when you're making yarn on a loom, the yarn starts to wrap around this needle. And eventually, what you get is something on that needle that looks football shaped. So it's almond shaped. So when you see spindle is appearing, look for an almond shape, because that's what they're talking about, the actual physical shape. So if you look at the early prophase model, look at how thick that genetic material is getting. See that spindle formation? There's my almond right there. Here's one aster, here's the other, and these are the centrioles that are basically pushing away from each other to get to opposite sides of the cell. Nucleus is still intact in this particular model, but the chromosome is being formed. You also have the cytoplasm, the GUI inside the cell, and the cell membrane. So you've got all these things that are in this particular early prophase model. Now looking at the late prophase model, by this point, the centrioles are on opposite sides of my cell. They're at the north pole and the south pole of my cell. Remember what I said about I've got to be able to split it in half? That means if I'm going to split it in half equally, I have to be on the opposite sides of the cell. The chromosomes are now being pushed all over the place to try and get them into place so that I can split them apart. The asters are still there. If you look, there's still that starburst pattern on both sides. It hasn't gone away. These red lines are actually the mitotic spindle. They're those microtubules that are pulling and pushing to get the DNA where I want it. Still have cytoplasm in here, still have a cell membrane out here. Now, looking at metaphase, meta, middle. Okay, so when you see meta, P-mat, metaphase, think middle. During metaphase, all of my copies are lined up on the equator of my cell. The chromosomes all line up at that equator. These centrosomes are basically pushing and pulling until I get everybody lined up in the middle, okay? The mitotic spindle, these are going to attach to the centromeres of each chromosome pair. It's like grabbing a right arm and a left arm of somebody that I'm going to split in half, okay? And you've got it clearly identified by the fact that you've got this equator housed chromosomes. You know metaphase more than you'll know anything else because of where the chromosomes are. If they're dead center in the middle at the equator of the cell, you know that it is metaphase, period, end of story. So when you look at this model, you can see the chromosomes are dead center in the middle. I've got one kind of coming up this direction and one kind of going down that direction. I'm lined up dead center in the middle. You can also see the spindle. There's my football, dead center in the middle, right? So that's the mitotic spindle. I've got my asters with my centrioles on the opposite poles of the cell. Still have cytoplasm. There's still cytoplasm in my cell. Still have a membrane. That has not changed. Now, anaphase. So once I have these guys lined up, I want part of it going this way and the other half going this way. I want the instruction manual going in both daughters, right? So in anaphase, I actually yank these two copies apart from each other and start dragging them toward the opposite poles. So when you look at anaphase, instead of having them attached like that anymore, I've broken this apart, and now I've got half going this way and half going that way. So when you look at anaphase, you'll know it's anaphase because these chromosomes have literally been split in half. And you can see they're copies of each other. They're the same size on each side. It's just that I only have one copy instead of that pair that was joined together before. So the centromeres are broken and the two chromatids of each chromosome separate. They're pulled to opposite poles of the cell. One's going to the North Pole, one's going to the South Pole. One set is going to the North Pole, one set is going to the South Pole. And it's caused by the pulling and pushing action of the mitotic spindle. Remember that these guys are still attached to them via these tubes, okay? Now, in reality, anaphase is actually rarely seen because it's like this split second of boom and it's done. So catching that on a slide or something is actually fairly rare. At the tail end of anaphase, you'll actually also start seeing the beginning of a cleavage furrow. Remember, this is going to be two cells. So it starts to kind of indent in, basically more and more, making it so that it'll split in two. That's the cleavage furrow. So this is early anaphase. They barely just got away from each other at the equator of this cell, but you can see that I've got half going this way and half going that way. You still have the asters. You still have the mitotic spindle, technically. It's still kind of a football shape. Okay? You have the centrioles. Admitted they're on opposite poles, but they're still there. Still have cytoplasm. You still have the cell membrane. But with anaphase, you can see that half is going that way, half is going this way. I've split them apart. Now this is late anaphase, okay? So now I've actually really started dragging these toward the North Pole here and toward the South Pole here. See this right here? This kind of indentation? This is the cleavage furrow. Imagine that I've got a tube of Jello in front of me, and I take a piece of dental floss and I start making it tighter and tighter and tighter right in the center of that jello. Eventually, it would split it in half. And that's exactly what's happening here at that cleavage furrow. It starts getting tighter and tighter, like the drawstring of a trash bag. And as it meets together, the trash bag closes, quote unquote, it actually splits so that one cell goes this way and one cell goes that way. So you've got the sister chromatid splitting, you've got the mitotic spindle, you've got the centrioles, you've got the cytoplasm, you've got the cell membrane, and you've got the cleavage furrow. You know late anaphase because the cleavage furrow, you can still see those sister chromatids being dragged and bending from that dragging. Now, the last phase, Telophase or Telophase. It depends on who you learned it from. I say Telophase because that's the way I learned it. Telophase is when the cytokinesis portion of mitosis happens. Cytocell kinesis split. So remember that cleavage furrow? We're really starting to divide these cells in two, or this one cell into two cells. Something else that's a big hint. The nuclear membrane starts to form again. Remember, we dissolved the nucleus at the beginning? You're actually going to see signs of that now. The nuclei has a new set of chromatin. Remember, half went that way, half went this way. So that new set of chromatin is going to start getting put into a brand spanking new nucleus in this new cell. That cleavage furrow continues to constrict until you have two separate cells. Again, the jello with the dental floss. That process is called cytokinesis. When you look at this model, notice there's a nucleus. Yes, we've got the cleavage furrow. Yes, it still looks like the sister chromatids are still being pulled, but that nucleus is the big, huge clue. Hey, this is telophase because the nucleus is starting to reform. You have the centrioles. You have the mitotic spindle. These guys still making a football shape. Well, half a football now. The aster, you still have cytoplasm inside the cell. You still have the cell membrane. Telophase is pretty easy to identify because that nucleus starts to show up again. Once that nuclear membrane starts to form, you know, okay, I'm in the last phase. Even if the cleavage furrow hasn't finished and cytokinesis hasn't started yet, or hasn't finished yet, hasn't concluded yet, it doesn't matter. The minute that you see that nuclear membrane forming, boom, you're in telophase. So, at the end of cytokinesis, you have two separate cells. You can see a membrane here and you can see a membrane here. They're separated from each other. Here's the formed nucleus with my chromatin in it. Notice it's not chromosomes, it's chromatin, because by this point it's unraveled. It's not that condensed version of DNA anymore. I've got one centriole in each cell. I still have cell membrane. I still have cytoplasm. That hasn't changed. But nothing is dividing anymore. I don't have two copies of centrioles that are going to start prophase or anything like that. These are just two new daughter cells.